All right, thank you for the introduction, Mike. We're going to invite our panelists up to the stage now. We've got some really impressive stories here, and we're going to encourage you all to come up with questions because we're going to leave a little bit of time at the end so that you guys can ask the questions that are most important, because ASOs are a big thing that are going on in the rare disease space in general, but especially in Dup15q. You're going to hear some specific stories later, but these are some of our experts in experience. And so we've got 3 panelists on stage, and we're going to have one that's joining us remotely, which is an amazing commitment. So I'm going to have our panelists take a minute to introduce themselves, and I'm going to start with Crystal, who's on Zoom logistically, so that she has an opportunity to introduce herself before we go to the panelists that are in person.
Hello, my name is Crystal Byrd. I'm from Kentucky. I have a 17-year-old son with Dravet syndrome, and I'm pleased to be here today and answer any questions. We've been in an ASO trial now for 4 years.
Is this working? I haven't been up here yet. James Fink. I'm a former Dup15q Alliance fellow and now currently lead an ASO program at a company called Quiver Bioscience, and we'll get to present some of that later. Looking forward to that and happy to be here.
Good morning. My name is Allyson Berent. I am the Chief Science Officer at the Foundation for Angelman Syndrome Therapeutics, and I was the co-founder of a company that developed an ASO for Angelman syndrome called Genetics Biotherapeutics that was acquired by Ultragenyx Pharmaceuticals. There seems to be feedback. And I'm actually also here not only to talk about the development of that, but also the fact that my daughter, who lives with Angelman syndrome, has been in an ASO clinical trial for over 5 years.
Hi, it's me again, Yael Weiss from Aussie Therapeutics, developing multiple ASO programs for multiple disorders. Hopefully one day Dup15q.
Thank you to our panelists for being here. I think we've heard the term ASO, or antisense oligonucleotide, thrown around a lot yesterday, and we're going to hear it a lot more today. So could some of our panelists maybe set the stage as to what is an ASO and what does it actually look like and how does it work?
I can speak to that from a parent perspective, because I think that's probably going to be the best perspective, and maybe you could speak to it more from a technical perspective. So an antisense oligonucleotide is an artificial piece of basically genetic material like DNA and RNA, and it's meant to modify the genetics in the cells. So in the instance of Dup15 or Angelman syndrome, basically you're taking a genetic construct that's made in a lab and it's artificial, like a medicine, a chemical, you're putting it into the spinal fluid. It actually has an unbelievable ability to get into neurons of the brain, so the cells of the brain, and it targets the genetics that are abnormal because it's like a scalpel blade, it's sequence specific. So it'll target the UBE3A gene in your case, and it'll target every copy of the UBE3A gene to try to actually knock it down, try to lower the level to a point that's been titrated in the lab and in animal models that would be ideally perfect to express the right amount of UBE3A. It's a fine line of not being overexpressing UBE3A protein and not underexpressing, because I assure you, you don't want Angelman syndrome. So you really, I promise you don't want Angelman syndrome. So you want to find that happy balance where it's closer to normal, where that will allow you to improve the symptoms of Dup15 that are related to the overexpression of the UBE3A protein. So it has to be repeated in dose because it only lasts a certain number of weeks or months. And so that's why you have to do a spinal tap. Actually, you have to put the material in the spinal fluid. In the case of Angelman, it's every 3 months. In order for that to get to the brain, it lasts approximately that period of time so that you're constantly keeping this period of knockdown of the, in your case would be the UBE3A overexpressed gene.
That's right. I'll add one thing, and that was great, Allyson. Thank you. I really appreciate that explanation. For any of you who have watched CSI or like crime dramas, you know, we can identify the suspect based on their DNA. You know, genes themselves, like UBE3A, have unique sequences that identify UBE3A and make UBE3A that particular molecule in the brain. And so these ASOs, you can design them so they are specific to that gene. Just like UBE3A has its own fingerprint, you can make an ASO such that it's specific to that gene and it's not going to necessarily impact other genes in the body.
All right, panelists, I'm going to ask each of you, you have different experiences with ASOs and successes in that realm. So would you each be able to describe the ASO therapy that you're working on or associated with, and what an early success of that ASO program might look like to you?
I can start because we're in the earliest of the ASO phases of the people up on the stage, and we really actually look towards groups like Allyson's for hope and the path that's being laid by them that we hope to follow in. So right now, our ASO for Dup15q is being tested for safety, early safety studies, but the sort of later safety studies are quite expensive and time consuming, so you want to really be sure that we have the right medicine to move forward to sort of further characterization. So that's what we're doing right now.
So generally when you develop a drug like an ASO or even a gene therapy, there's many steps you have to take to get from what's good in an animal to what's good in a human, and then is it actually safe and is it working in a human? And so those steps the first step is the preclinical or the non-clinical aspects of that. So it's selecting the right drug, the right candidate, and then it's moving that candidate or a group of candidates to the animal studies. Does it work in a mouse? Is it safe in a monkey? Does it work in a monkey? In some cases, there's the sequence of UB3 in the monkey and the human is really almost identical. So you can test actually the knockdown of dropping UB3 levels in a monkey. And that sounds crazy, like we're doing experiments in monkeys. Monkeys have really helped to shape how we understand dosing. What dose do we need? How frequently do we need it in order to be able to get to humans? And then you submit all this information, which is years and millions of dollars of information to the regulatory authorities like the FDA. And they say, okay, based on what you're showing me, there's plausibility this could work. And there's safety behind it. We've done the right thing to show it's safe in animals. So we give you clearance to try this in humans. But I emphasize the word try, because what that means is it's an experiment. And whether we like it or not, the best experiment of all is that of our children. And that doesn't sound great, but the only way we know if it's going to be safe or it's going to work is to try it in humans, because that is the species of choice. And we don't know sometimes what the reaction will be in a human versus what the reaction is in a monkey or a mouse. And so that phase 1 clinical trial, once the FDA gives you permission, is to test it in humans. Number 1, for safety. Is it safe? Okay, it is. Number 2, is it doing what we want it to do? Is it knocking down UBE3A? The only way we might know that is if there's ways we can test that, what we call target engagement. Is it causing UBE3 levels to go down? Well, we can't do that really in humans unless we do a brain biopsy, right? And we're not doing that, I promise. So then we're looking for other downstream ways to look. The CSF fluid. Is the UBE3A high, and are we making it lower? That's something you're looking at in your natural history studies, maybe, of collecting CSF fluid to assess that. Do we have high levels of UB3A? The EEG, was it abnormal? Is it more normal? But maybe you're going to say, my kid was having 50 seizures a day, and now they're having 1 a week. Somebody's doing something for your kid that's really promising. So these are all the ways that the phase 1-2 early trials will help us determine if it's working. From there, we say, okay, this is what we saw in the kiddos, and these are the primary things we're going to look at, because we believe with conviction that these endpoints are promising for this population. That's going to drive how we design the next final study, which is called a phase 3 or pivotal study. That is the study used by the regulatory agencies to approve a drug. So you have to go through all of that before the whole population can actually get the drug. So where we are, so they're in the non-clinical, preclinical stages of those safety studies, which is still doesn't, it seems low on the totem pole of timeline. It's actually really far ahead because years went in to get there, right? So the biggest timelines are early discovery. They get tighter as you go along. So now they're in a tight timeline, probably only, a year or 2 to get that information to go to first in human. The first in human studies take maybe, 1 to 3 years. And then the pivotal study can take 2 to 3 years. And so where we are in Angelman is we went through all of this process from 2017 to 2019. We dosed our first patient early 2020, a week before the pandemic really, actually the first patient dosed March 10th. New York City closed down March 13th. So it was a really good time to start a clinical trial. And then we ended the phase 1/2 with positive data in 2024 and started phase 3 in 2024, expected to be done with that by 2026. So where we are right now is in phase 3. So what that means is we had positive clinical data to drive endpoints for phase 3, and it was safe enough for the FDA to agree that the dose in which we can move forward with is safe for humans. So that's where we are. We're in the phase 3 part of the clinical trial landscape.
So I wanted to highlight Spinraza, which is actually the most famous antisense oligonucleotide that's already been approved and has been used in many children with SMA, spinal muscular atrophy. It was not developed by us, but I do want you to know that there is an antisense oligonucleotide that has been approved, has been in the clinic for many years, and has really been transformative for SMA. And this was the antisense oligonucleotide that led the path for many other programs to start, because had it not been successful, then people would have said, that's not a great platform. But because of its success, we're able to start developing many other antisense oligonucleotide programs. And just to add on to what Allyson said, and Allyson also mentioned yesterday that a lot of drugs start the path of drug development but never make it to be a commercial drug. And there's so many ways that a drug can fail, and so many stations along the development path that it could prove to be not what we hoped it would be. So you also need to understand that you having multiple shots on goal is actually an advantage. Having several companies and entities developing an ASO guarantees you more shots on goal. The reasons that they can fail is, as Allyson just said, we start with mice, we start with monkeys, but mice and monkeys are not humans. So even if we showed really good, Allyson, you always say, many mice were cured from Angelman, right? But not people. So translating from a mouse to a human is a big jump, right? We cured a mouse, that doesn't mean we can cure a human. So first of all, does the efficacy that we see in a mouse translate to humans? Is the mouse behaving better? Does that mean that the human will show different behaviors? And then there's safety. There are always concerns about what we call off-target effects. Does the antisense recognize other sequences in the body? Does the antisense have any tendency to do something that it shouldn't be doing? And this is what we test in human clinical trials. And sometimes we find that the drugs are not safe and we have to stop the programs. So the path to making a drug available for all of you is very long and very risky. And again, this is why having more than one shot on goal is really an amazing place to be.
All right, I'm going to give the families that have had experience with an ASO treatment for a son or a daughter to talk about what that meant for them.
I think that's one of us. Actually, no, we have someone on Zoom. Why don't we give you an opportunity?
Crystal, we're going to give you the
Kristen,
opportunity to start off.
Well, I was very excited. Our time to try the trial was during COVID, so it was very emotional for us because our trial got prolonged, it got put off, and we had researched, and we've been very excited to get into the program. And when we finally got to go in, our first injection was not as great as it had been planned. The anesthesia, he got very sick for a few days, but he recovered, and then we tried the second time, and then things started getting a lot better, and it was one of the best decisions I think that we've made for him medically. And we've been in a few trials now, but at first it was very scary because no one really knew any answers to tell me. They were just using the mice and the monkey studies and giving us that data. But my neurologist and our epileptologist was very positive with me and went over the mechanisms and things that would be going on in his body. And we decided that was just something that we needed to try because he needed he needed more. He was having 6 to 8, I think, seizures at that time, mostly nocturnal. And now he is about 1 a month, maybe. I'm sometimes not even 1 a month. So it was a good decision. It was hard, but it was just something that I don't know. I felt that we needed to do.
So for Angelman syndrome, it's not necessarily as severe of an epilepsy as Dravet or Dup15. So our kiddos generally don't have refractory epilepsy, where some of them do, but very, very rarely is it refractory to medications and response to medications. Doesn't mean we don't want our kids off of seizure meds, because they are almost all on seizure meds, but they're generally pretty responsive. So a little harder to actually measure success, because not that we want to dream for seizures for our kiddos, but you can count them. And you can see them on an EEG, and you can count them every day, and you can have a very clear path and graph of what success looks like. Because obviously, if your kid is seizing less, their brain is improving. They're not having those firing episodes that they would be having otherwise. So with Angelman, really the successes had to be tailored to understand those endpoints in a really unique way, more about developmental milestones. Is my child able to sleep? So our kiddos, some of them sleep 2 or 3 hours a night. But not all of them. Some of them sleep 9 or 10 hours a night. So how do you measure sleep improvement if your kid sleeps well? Right? So we had kids that were sleeping 3 hours a night. And let me just say, there are 3 ASO clinical trials in Angelman syndrome, 2 of which are in phase 3, which means that over 300 kids, or nearly 300 kids now, have been dosed with an ASO. And the phase 3 clinical trials, each study is over 100 individuals that are included. 200 in one, 150 in another, and 120 in another. So if you look at the totality of the people that are going to be enrolled in ASOs for Angelman syndrome, it's going to be near 600 people. And so I'm just speaking about the totality of the publicly available data from each company. And then I could tell you my own personal experience is that individuals that were sleeping 3 hours a night, there are many that are sleeping 9, 10, 11, 12 hours a night. Parents are sleeping in the same bed together, having not slept in the same bed together since their child was born, because our kiddos like to sleep with their parents or their siblings. And so now they're sleeping independently in a bed. That's huge for our community. Kiddos that couldn't use their talker, their iPad to communicate, have finally an isolated finger point, fine motor improvement, but they actually can now communicate. And we had no idea what they actually were trying to say for so long. And they're communicating with their iPad. Kids that didn't have even one symbol, having 50, 100, 150. Kids that couldn't self-feed, that are self-feeding. Kids that were having some challenging behaviors, having less impulsivity and more calmness, being able to attend and focus on tasks at school, being able to play with classmates without grabbing and pulling and mouthing objects and things like this. So these things that are a little harder to measure are just showing up in all 3 trials. And it's hard to justify that it's placebo effect when you're seeing it in all 3 trials. And I think the most profound thing that I've seen are kids that are independently swimming. And I think I said this yesterday, but in Angelman syndrome, it's considered a non-degenerative disorder in which there's not a high mortality rate, because our kiddos generally don't have major seizures, but they can die of seizures. But one of the leading causes of death is trauma. The leading cause of that is walking into a pool and drowning in the middle of the night, because kids love water and they get out and they get into their pool. And that is devastating. That is absolutely devastating. And to see a child swim independently, I'll cry, without a floaty, to jump into a pool and be able to get out, means that child might not die. That means the parent's not afraid to build a pool, which is the happy place of that child, because their child may not die if they get in that pool. That to me is something that you can't measure. It's not on the Bailey, it's not on the Vineland, it's not on the CGIs, it's in video, it's in interviews. That transforms a family's life, that they can take their kid to a beach and not worry they're gonna walk out to their death. And those are the things that people are seeing and talking about. And I can tell you from my family's perspective, I think one of the best examples we have is that my daughter's potty trained. That's not something I ever expected for her. And she's 11 now. An 11-year-old in a diaper is not as cute as a 2-year-old in a diaper. And for her to be able to apply for camp because the inclusion criteria was she has to be potty trained, changed our lives, because she can be in camp with friends. She can be without a caregiver in camp because we would have to send a caregiver with her to change her diaper because the counselors won't do it. That's independence. That's her being in the world in a place in which she wasn't included, all because she now knows how to use the toilet. This is something we dream about for our kids. So these are the little things that give us so much optimism and hope, because kids with my daughter's genotype of Angelman, large deletion, they're not usually potty trained. They don't usually independently swim, and they certainly don't talk. And my daughter has words. My daughter can say cliche, but she can say mama. She can say baba, which is her father. She can say Piper, which is her sister. She can say come when she wants us to come to her and doesn't pull our hair now. She doesn't grab us. She calls us. That's huge for us. But what does the Vineland measure? We've all taken the Vineland test, right? You get a movement in speech if you have 50 words or more. She doesn't have 50 words, but she does have many words. And those words have changed her life, and importantly, have changed our life. She has a relationship with her sisters she didn't have because she can call them. She can ask them to play rather than just steal their things. That's a relationship she didn't have, and our life is interactional now, not so transactional, where we were doing things for Quincy. Now she's doing things with us, and that is a completely different shift in relationships, which is all we want for our kids, is to have a happy life filled with relationships and connections. And so for me, it has transformed her life in the hugest way.
I think we could all tell those were some pretty powerful stories there. The voice of the families is so important, and I'm curious to ask, kind of on the pharmaceutical or the biotech side, how do you guys interact with families to understand what's important and how you can measure success in a clinical trial?
I can start. Every time we decide to take on a program, and we do it based on the fact that there is a strong community that has the desire to work with us to move a program forward. And the first thing we do, especially if it's a community that has not had a therapy before, is ask them what will be a meaningful change for you. So in a seizure disorder, everybody thinks it's a no-brainer, right? You want to reduce the number of seizures, but in many cases when we interview families, we find that there are a lot of other things that families want to see changed, just like Allyson was just talking about. Swimming, speech, sleep. But when you actually dive deep into the family's desires, you find things that you originally did not think about. And you may want to design your clinical trial based on what you hear from families and not necessarily what your preconceptions were about this disorder or the symptoms. So we do very deep surveys and interviews with families to understand what meaningful change will be for them. And then we design our clinical trial accordingly. So for example, we're working on another neurodevelopmental disorder called Pitt-Hopkins. When we interviewed the families, we found that the number 2 concern for these families was GI symptoms. And we would have probably placed GI in number 4 or 5 because we thought seizures and speech would probably be the first 2. So speech was the first, GI was the second. So now our clinical trial is designed in such a way that we measure GI symptoms very carefully, and we may not have done so if we had not interviewed the families and found out how important it is to the families. So it's critical, and you'll be asked to probably participate in surveys. You'll be asked to talk about what's meaningful for you. Obviously, we harped about the natural history study yesterday, and I think Dylan will talk about it again today. All these pieces of information come together to design the clinical trial that will measure what you need and not what someone thought you needed. So it's really important to have your input, and your input will be critical for what a product will look like at the end of the path.
And I can say that I'm very privileged compared to some of my peers in graduate school to have been able to attend the Dup15q family conference year after year, starting from 2015. And there's definitely a perspective that offers that you don't necessarily get when you read papers in the lab. We're a small company at Quiver, and I can tell you that hearing stories from families changes the motivation for why we go to work every day. And that's been really, really important to how we get up and go to the lab, and we know we're working for the families in this room, not necessarily assess symptoms on a piece of paper.
And with that, I want to give the families an opportunity to come in and ask a lot of our experts. This is something that's coming up in our field. We're going to have 2 great talks today from our ASO partners. One of them will be run by James, but we have a unique opportunity to ask families who have gone through the process, and also some of our industry experts, what that might mean for them. So I see a hand up over there in the audience.
Thank you so much for being here today. One issue that you were saying about, as far as having feedback from parents and families, that we were just talking to some pharma people about, and I'm really curious whether this has come up with Angelman's and your trials. We're seeing cases. Initially, I was always so afraid of seizures as the reason that we could lose our child. But we are seeing children with Dup15 as they age into their 30s or even in their 20s having so many issues with swallowing and choking on their own secretions, which can worsen, and then they keep getting pneumonia over and over. And I have been speaking with our this is a neurological problem. It happens with people with dementia frequently. At least that's my understanding, that someone with dementia, they start having problems swallowing, and then they choke on their own secretions, and then they get pneumonia. And I've seen this with another child with Dup15 at our school. Is this something that you see, and is it anything that's being targeted with the Angelman's trials?
We don't see that in Angelman. As they get older, they get other challenging issues that changes with age. Even though it's non-degenerative, some of their skills change, particularly related to motor function for us. And with non-epileptic myoclonus, so more of a non-epileptic type of movement disorder that our kiddos get as they age. But swallowing issues, Ron Byrd is here, so we could ask him because he's the expert on both. And so he can probably comment on that. But I haven't seen that in our community. And so it could be an excessive UBE3A problem, not a lack of a UBE3A problem. We always joke, we have good friends who have a child with Dup15, that if our kids could only get together and make babies, they'd be perfect. But we don't see that in Angelman. So it's not targeted.
Well, one of the things that the information you'll get if you enroll in Citizen and in a natural history studies will be information about hospitalizations and why patients are being hospitalized. Is it because seizures? Is it because of seizure complications? Is it because of aspirations? And then when a drug is introduced, we can follow to see if there's a reduction in these hospitalizations. And it's really important not just for you as families to see a reduction in hospitalizations, but also when we go to a payer and say, listen, this child is costing you a lot of money because it's hospitalized 5 times a year or 10 times a year, and our drug allows for only 2 hospitalizations or no hospitalizations a year because of aspirations, then this will help to build the case to actually get this drug reimbursed by payers. So I know this doesn't answer your question directly, but it's something that definitely will be measured and followed in a clinical trial.
Thank you so much.
We have a question from the left side of the room over here.
Yeah, question. What happens when the gene therapy trial ends? Do they need to be on ASOs their whole lives, or?
So I think the question is what happens when it ends, meaning when it's canceled or when it's completed.
When it's completed?
So different things can happen. So it's really up to the company. For example, the phase 1/2 trial completed and a new trial, phase 3, started. It's not just a continuum. And it continued because there was positive clinical data. So this is the example for both Ultragenyx and for Ionis. On the other hand, we had a third trial, which actually went through phase 1/2 by Roche, and they decided not to advance the program into phase 3. So the trial ended. And then another partner came in, thank goodness, at the midnight hour and picked it up. And so it will make its way into phase 3 now, but that interim and that gap when it canceled versus it reinstituted, kids went off drug. And if the trial ends without being continued in the clinical trial phases, then the drug is discontinued as well. If the trial continues, you go from phase 1/2 to phase 3, most companies, I believe morally and ethically are responsible, if they're responsible, will continue on a long-term extension. What that means is they will continue giving drug to everybody that started the trial until an approval or a failure. If there's an approval, everyone stays on drug, but now you transition from being on the ASO for free in a trial environment to actually your insurance company needs to start to pay for it. If it fails and it does not clear regulatory hurdles, then ultimately everyone goes off the drug because it's no longer available, or a different company tries to do something with it. But it really is regulatory dependent, it's financial dependent, and it depends if it moves forward from a company prioritization perspective to continue to fund it. Does that help answer that question?
Because we have less than 10 minutes left, I think it's worth also mentioning, we talked a bit about this, the science breakout yesterday, but there's also a theme during the investigator science conference earlier in the week. And I want to sort of quote as best I can, Mike Ehlers is a sort of stellar scientist and now venture capitalist who's very involved in starting lots of companies. And he was describing the starlight effect in science, which is that the technologies that we're using today in the clinic are those that have been being developed, as Allyson mentioned, over the past 5 to 10 years. But newer technologies are much earlier and maybe even more promising. And so one of the talks we had heard was from a company called Denali that's now actually developing ASOs that you can give intravenously. Now this is far away from being an Angelman and Dup15q, but much closer than we might think. And I also think there's recently some news about a new version of Spinraza that is going to be longer lasting. So anyway, this is, I think, the current technologies that we're talking about, those are the ones closest to being in patients and are in patients. And so we should be extremely excited about those. But I don't necessarily think that the medicine that those patients are on today are actually going to be the same medications that they're on in 10 years.
Yeah, I totally agree with that. And I think it's important to also mention it's this balance of the enemy of good is better, but you need to ensure you understand version 2, version 3, version 4. We didn't start with, Aleve and COX-2 inhibitors for inflammation, right? We started with like aspirin and like really basic anti-inflammatories. And the technology moved to make them better and safer and with less side effects and to last longer. That's exactly what's happening in gene therapy, in cell therapy, and in ASO therapy. So we have to realize if we wait for the best, we're going to get nowhere fast because it's always going to get better. So we have to start somewhere. We have to start with proof of concept. Does this work? I would argue in Angelman, we've shown this can work. But how do we make it better? What about this is hard? Going in under general anesthesia, our kids need general anesthesia every 3 months for a lumbar puncture. That's hard. It's hard for them, it's hard for us. It takes a lot of time, and that has its own risk. If we can go IV like Denali and just go have a port, and a nurse can come to the house and every 3 months inject IV, that'd be amazing. We wouldn't have to go anywhere. That would improve the delivery to more of the brain, and it would improve the convenience, and it would improve the fact that my daughter wouldn't have to miss school, right? So every step is going to get better and better and better, but if we don't start, we'll never know. And this is where people get stuck. Everyone wants to know more, learn more, understand more. Guess what? Most things in our life we have no idea about. Why do you take supplements every day? Because it doesn't hurt, might help, right? My daughter's on like 15 supplements. I don't know what almost any of them do. But in a mouse Coenzyme Q10 did something to the mitochondria, so give it a shot, Quincy. Like, you just try stuff because it won't hurt and there's nothing else to do. And so it's important to not get stuck in the tomorrow. We have to live in the today, but don't give up and don't stop funding the next versions because they will get better and better. And if you wait and don't fund that once you have it today, then you're going to be behind, right? So it's this balance of like, we need to get something there, but we also need to keep our eyes open to amazing technology that will make everything that's today better. And I think it's hard to balance that at times.
We have a question on the right side of the room over here.
It's kind of a question. It's kind of sharing information. I have 3 children with Dup15q syndrome. 2 of them have an identical duplication, but he is mosaic for Angelman syndrome. When we got the genetic, they thought he would be our most severely impacted, and he's really high functioning. Any useful information with that? Would that be helpful for what you guys are doing?
Oh, you are a case study. That's amazing. Yeah, you know, mosaicism has been one of the most helpful things we have seen in Angelman in terms of telling a story about what's possible, right? So what we know about ASOs is they literally probably get to less than 20% of the brain when they're delivered by CSF. If they go IV, they probably get to 60 to 90% of the brain. So when you look at a mosaic individual, they can have different levels of mosaicism. So what percentage of the cells in the brain are expressing UBE3A on the maternal allele? And what percentage of the cells are not? And we have a couple of really older studies where you can see 1 to 5% of kiddos, 5 to 10%, 10 to 20%. Angelman is not diagnosed over 40% of mosaicism. And that's because they're considered neurotypical probably, which means to us, you probably don't need more than 40% to be near neurotypical. But what it also means is that a 1 to 5% kid has words, walks normally, and has no seizures. My child is 0%. I would take 1 to 5% any day because that is a different child than I live with. So your kiddo could have 30% mosaicism. That could look close to a child maybe with a little learning disability.
Well, he's Dup15q and Angelman.
So
he's 80%
he's giving himself back what he's missing. That's
yeah,
amazing.
that's kind of, and he functions almost typical. That's why I was wondering if.
So I suspect you gave yourself a gene therapy right there.
Yeah, that's what we were wondering. I just didn't know if it'd be helpful for what you guys are doing.
That's very helpful.
Okay.
And actually quite amazing.
We have a question on the left side of the room over here.
Yes, for the virtual parent, we'd like to know if you've had any other improvements besides seizures. And also, with each application of the medicine that these patients are receiving, do you see an increase with each application, or is it pretty steady as far as everyday living improvements, like with your daughter? You know, is she maybe getting 10 more words with the next application?
In my instance, we started out with a very low dose at the time because they weren't allowed to give us a high dose of study drug. The initial dose was 10 milligrams, I think. But anyway, he did really well. We have seen he speaks now. He can communicate in full sentences in first person most of the time. He can swim independently now. He's always been potty trained, though. Oddly enough, he doesn't like to be wet or soil himself. So that's probably why he's fully trained. When we upped it to 20 milligrams, we titrated up. We did see a lot more engagement. He wanted to go out into public. He wanted to be in large groups, and he's never wanted to play with kids. He would watch from the side. Now he wants to join in. He would try to I guess, communicate with people to get them to come near him, which is very odd because he never did that before. Then we are on the final dose now, and most of his neuropsych, the people that have done it, said that they've never seen such a quick, it's just almost like a different person. I don't know how else to say it. I don't want to use the word normal because I don't like that word, but if you knew Charlie 4 years ago versus now, you would think he was a new child because he is completely different. And it's hard sometimes for people to even know that there's much going on with him. I mean, you have have to really pay attention, but it's been, and he sleeps better now. He sleeps way better than he used to. That's another thing that they don't ask you when you're, in the tests, but they always, and his GI issues, which I give him a probiotic and a prebiotic, so that may help, but those have improved. Trying to go through the things. I don't even know how to quantify how many systems have positively been impacted. So it's just really hard. But it's been wonderful for us. And as a married couple, I can sleep with my husband again. We're not, we still sleep with him sometimes. It's because he's my baby, but we can now have a normal family dynamic. And he goes his sister's softball games. He sits through the whole game, goes to church, prays. It's odd. I don't know how else to say it, but it's been wonderful for.
And the difference here is, like, these are real stories, right? These aren't stories that are in a graph, in an industry presentation, where someone's saying the median improvement in the gross motor skills are X, Y, and Z, and the cognitive score is better than natural history data. That's all important for the regulatory authorities, and that's important for investors. But people are reluctant to take personal stories like this, that some of this is not measurable. Some of this you have to feel and see and live, in which case the regulatory agencies don't, investors don't, and industry often doesn't. Not because they don't want to, because they can't. And so telling these stories is super important. And to answer your question, my daughter, I would say, there's a load at the beginning of a dose escalation, and then there's a plateau of dosing. And in the load, it was exponential. Like within 10 days, she was walking independently up and down stairs with reciprocal steps without any help. And she needed 2 hands on a rail and an adult behind her in front at all times. Now she's independent on the stairs completely. I saw that in 10 days after the first low dose. And every dose that went up, I don't know if it was the higher dose or just the fact that there was more time for learning, you saw her gain more and more skills. By 3 months, she had her first word. That accelerated to about 27 words within, I would say, about 5-6 months. The words have plateaued. She's staying there. Sometimes she has new words. Sometimes she doesn't use an old word for a while, and then we'll just use it again. Her sleep has always been pretty good, but it's better. So she was always an 8-9 hour kid, and now she's a 9 to 11, probably a 10 to 12 hour kid. Sleeps solid. She used to be very restless, things like that. But I would say that the gains in plateau are on a trajectory up, but they're much slower and they're much less obvious. So, for example, the other day we were on an airplane and she looked at me and we were just descending and she went like this. And she's always happy. Angelman kiddos, that's their MO. They're like happy children. So she's smiling, laughing, playing with the spinny thing. She was telling me her ear hurt. Our kids can't tell us what's wrong. Her ear was not popping. And I said, does your ear hurt? And I gave her her water bottle. I said, sip, this will help. And she sipped. So she's following instructions. She's expressively communicating. She's touching the body part that she knows hurts because she knows mama will help. Or she needs help, she takes my hand and puts it on what she needs help with. She goes to her talker and says, help. These are new complex things that she didn't have before. Mm-hmm. So I don't know if the dose is making her better or just time to learn. Kids don't say their first word till they're 1. Our kiddos had their first word at 3 months. Like, that's crazy because we expect learning takes time. So I think that it's more that she is learning, but that trajectory went like this and now it's like this, if that makes sense.
I hope the families can understand why we at the Alliance are so invested in supporting our ASO programs, and it's not because there's no support or there's no logic behind it. We hear these stories from a lot of other groups who have seen successes in the clinic, and the successes at home may even outweigh those that are quantified in the graphs. And so I hope you guys understand and pay attention to some of the ASO talks that are going to be going on today. But I want to take a moment to thank our panelists so much for sharing all of their wisdom and experiences with you all today.
You're so welcome.
Please take the opportunity to track them down after this and ask them questions because they're so experienced personally and professionally, and they're here because they want to interact with you guys. Also, this is a plug to participate in research studies so that we can understand what you guys are going through, so that when we get to clinical trials, we have an understanding of what you guys are looking for. So thank you all for listening, and we're going to move on to our next session now.